So yeah, it started. Of course, I had some more issues. Hello, welcome everyone. I think we're going to start. Just have some Zoom technical Zoom-ish thing. So welcome to this webinar in the FibRe webinar series that the Competence Center FibRe is doing together with the Treesearch. And Annette, I counted, I think this is the 25th
Wow.
webinar.
So we celebrate today.
Something to be proud of. And if you missed the previous webinars, most of them are found at Treesearch website. So today we will have a presentation then in the series, and if you have questions, I hope you have questions, you can ask them after the presentation. And you can type it in, you have Zoom, you can type it in the chat. You can raise your hand and we let you speak, or in any way doing that. If you're on YouTube, you can type in the chat. So I leave the word to introduce today's speaker to Annette.
Yeah, I can say I'm very happy to have a so nice speaker here today, but Per will be the one who introduce you.
Okay, thank you everyone. So yeah, today's speaker has been on our shortlist for quite some time, and finally we have managed to get him on board to give a presentation. So today we have the pleasure of having a presentation by Dr. Yoshiharu Nishiyama from CNRS in France. And I would say that Dr. Nishiyama needs little presentation within the cellulose community, and a community where he has actively contributed for more than 20 years. And among many things, he has been part of pioneering the field of cellulose nanofibrils at the University of Tokyo in the early 2000s, resulting in the Wallenberg Prize. However, for the last 2 decades, I would say the main focus has been on the scattering of ordered cellulose and unraveling the nanoscopic structure of cellulose, which will be the topic of today's presentation. So with that, I think I leave the word to you, Yoshi, and the screen is yours.
Thank you very much. So the title for today's presentation is Structure Analysis of Textured System. So Per, do you understand when I say textured system, do you understand what I mean? I mean, so that when I asked ChatGPT just textured what it means, and it doesn't, well, it gives all kinds of definition that I didn't mean. And here, when textured is combined with X-ray, ChatGPT can answer relatively well what I mean. And when I mean textured is you have some crystallographic preferred orientation in a material. Actually, it's very rare to have a material that has no texture. It's very difficult to have a material that is not textured, especially when it's cellulose-based. And well, this is extract from ChatGPT's answers, but it influences mechanical properties. Typically, for example, if you have a metal, rolled metal, then it's stronger in this direction. We can have, in case of cellulose, it is influencing swelling properties and, of course, make all kinds of mechanical properties. So texture is very important. And also it is studying texture is a very old stuff. So actually I have been studying X-ray scattering on textured systems since my undergrad. So it has been 30 years. And it's very simple, much simpler than people would imagine. So my presentation I would try to convince you that it's really easy and very intuitive to study textured system. So the first publication on X-ray scattering of textured system is 1913. Well, there's a typo in this printing, but you can just download from the internet the whole paper. And this is by a postdoc at that time, Dr. Nishikawa and Ono, who was it is just 1 year after the discovery of X-ray diffraction by Laue. And what they did is just put in front of the X-ray beam all kinds of substance that he finds in the lab, and look what kind of pattern he gets. So there's some examples like asbestos. Oh, I don't see my. So asbestos is a fibrillar inorganic substance that you insulate. Nowadays, you have to remove from the buildings, but it has just simply putting the asbestos sheet at a different angle, and you see interesting pattern. I come to and we celebrate. How to go to? Yeah. So here we have the first X-ray fiber scattering diffraction of cellulose. So we celebrated 12 years ago in the ACS as the 100 years of fiber diffraction of cellulose. It doesn't look like standard diffraction you see, because at that time people used tungsten X-ray and continuous X-ray. It was not a monochromatic X-ray. And that's also what Laue used to get a nice symmetric diffraction pattern. And you have an example of copper next to it. The system they used is very simple. So because they had nothing, there was no X-ray like Anton Par did not exist. And what you could do is to borrow. What they did is to borrow an X-ray generator. X-ray existed because people were doing X-ray body analysis to look at. So they had it in the hospital. So they borrowed a very old X-ray tube from the hospital and put a collimator, another collimator, and put the sample, and you have a box, and then you put a film, photographic film, and that's it. This is the whole setup, and you just expose for some time, and then develop what you see in the film. So this is what we have today in our lab, which is very close to what you see in the sketch for the 110 years old sketch. We have a X-ray. We have an X-ray lamp. This is the X-ray source. It's the same kind of tube. It's just a filament and a target. So that generate X-ray like a lamp. It costs about 3,000 euros, but it's just a lamp, and we have one collimator here and one close to the generator. So it's exactly the same as the sketch, and we have slits where we can insert the film. We used to use film until 20 years ago, and this is how we put our sample. We can put any types of sample on so you see a small hole, and behind we have another small hole with a lead pinhole. But you can put liquid or solid or of any shapes. You just put in the beam, and you can record. So nowadays, since 25 years, we use this kind of plate, which is called imaging plate, which we can reutilize many times. So this is a reader of imaging plate. So compared to photographic film that we have to have a dark room, we don't need the dark room, and we can just get the digital data quite easily. this has been popular for maybe 5-10 years, around 2000s, and now many people are shifting to the X-ray camera, digital detectors. But the advantage of this is that it's very low price. Each plate is about 100 euros, and you can use many times. And this system has been working for 25 years without any problem. So it's very robust and maintenance free. So that's why I like it. If you buy a detector, you have to invest a lot again in the maintenance. And here it is something that is really simple, that is robust over a century. By the way, the camera I've shown here, it dates back. This is brought from the United States in 1950s, and it is still in use because it's just a camera that you can pump in vacuum. The way we convert the X-ray that is stored on the imaging plate to digital data it's also a very simple system. You have a laser, red laser source, and you have a moving mirror that is scanning in one direction while the image plate is rolled in another direction. So you get a grid pattern of the intensity from the energy which was stored in the image plate, which emits visible light, and you have a photomultiplier to just detect the emitted light intensity. This is also very simple system that can be that is used in veterinary imaging or dentist. If you go to dentist, you have similar type of system. In many dentists, they have the similar system. Of course, you can also have a lot of more sophisticated, more expensive X-ray setup, like in 4MAX in MAX 4, or at ESRF in Grenoble. There are one in Hamburg. In many countries, you have synchrotron, and you can do similar types of experiment with much brighter beam or with more sophisticated detecting system. But essentially, what you can do a lot of things already with the simplest setup. So this is a publication we did maybe 15 years ago using our camera. So here what you see, it's just a casting of the cellulose nanocrystals and looking at different directions. So if you cast a film, you can put the X-ray either perpendicular to the film or parallel to the film, and then you already get different pattern. So this is well, I will explain it later. So just an example. There's also another very old picture which is very important in cellulose. This Sponsler published in 1931 on Wallonia cell wall. So Wallonia is a unicellular algae, giant algae. So here you see on the hand, each ball is one cell, and so you can handle the cell wall very easily, and you can pile up and put into the beam. And this is the pattern that can be obtained from the Wallonia cell wall. And this is directly related to the drawing here, that we have an image of the cellulose microfibrillary for a very long time, and which is just based on this kind of pattern, which is very simple, but you will see that, so you have spots of strong spots in a diagonal region, which corresponds to the about 4 angstrom spacing, which corresponds to the spacing between the parallel ring, and then you have one at corresponding to the 6 angstrom and 5.4 angstrom. So this pattern corresponds to this arrangement, and also another one in the opposite direction, the mirror image of this, which shows that in this cell wall, macroscopically, you have a preferred crystallographic orientation, not the chain axis orientation, but the plane of the cross section of the unit, the fiber, is oriented preferentially with respect to the membrane, and this will directly indicate a kind of shape of the building block in the cell wall without having using the microscopy, without seeing each individual structure. And this is another example that it's really perturbing to see this very nice data published in Nature Communication, but the interpretation of this data is completely wrong. So the title says preferred crystallographic orientation of cellulose in plant primary cell walls. And you see their reasoning is the following. So they did this kind of grading incidence experiment where you put a thin cell wall on the substrate and you shine X-ray from the grazing incident and detect the pattern on the detector. So they have 3 types of samples that all show roughly the same thing. By the way, those are peaks from the wax of the surface of the cell, and here you see the cell. I don't know if you recognize the strong cellulose pattern, equatorial pattern of 200 and 11011/0 diffraction. So they conclude from this pattern that there is a preferred crystallographic orientation like this. So the reasoning is that, okay, you see very strong 200, so some crystals are lying here. You also see very strong 110 and 11/0, so some cells should be aligned like this. Do you agree with this argument? So I wrote to them that they should retract this paper because this is completely wrong. And the reason why it's wrong, well, you can just see from, if this is the case, if all the fibers are aligned, you're looking only the section. But even in this case, if you had this kind of crystal, then the 11/0 diffraction should point to this direction. So you should see diffraction here, which you see, but only faintly. So if you had the dominant diffraction like this, you should see a combination of pattern like this. When you have a strong alignment of this one, the other peaks should appear in different direction. So I will try to explain how because maybe it's quite a little bit complicated to explain if people are not used to the reciprocal space. So we made a small paper with a student. So Xiaohan was a master student at that time, and it's kind of, she made most of the drawing, and it was really surprising for me what people were imagining about 2D diffraction pattern. So for her, 2D diffraction was something more advanced than powder diffractometer. But of course, 2D diffraction is the first one which was ever recorded, and diffractometer is much more sophisticated because you need some control of the orientation. You need goniometer. And so she was writing the draft, and I was correcting and trying so that she will understand what's happening in the pattern. So the drawing I'm presenting is drawn by her. And here what I introduce is the notion of reciprocal vector, because it's very so what when we say a certain crystallographic plane, we have a arrangement of atoms or a kind of variation of electron density that is periodic, and which we imagine a plane. But imagining a plane, a lot of orientation is very confusing, because, well, to draw one crystal is easy, but if you have a lot of crystal like this, it becomes a mess. So one way to represent a series of plane is to draw a vector which is perpendicular to the plane. And then the length of this vector is inversely proportional to the distance between the plane. And that's what we call the reciprocal vector. So then each plane, a series of plane, can be just represented by a point, the end of the vector. And then if you have a lot of crystals, then you can just look at the density of the points, which is much simpler to imagine in your head, or even for drawing. So how does it relate to the diffraction? This is the only maybe mathematical representation. So okay, this is what we usually do when you have powder diffractometer. You are moving the X-ray. Very often you have a sample, and either you move the X-ray and detector at the same time, or you rotate the sample and the detector at the same time, and you get just one-dimensional pattern. And this is what is usually in the textbook of the Bragg condition, where you have the Bragg condition is when the X-ray is entering at an angle theta and going out at another angle theta with respect to this plane. The difference of the X-ray coming here and coming to the second layer, the distance that this X-ray travels corresponds to twice the d spacing multiplied by sine theta. So this distance and this distance is the difference in length. And if this length is exactly at the wavelength, they are in the same phase. Whatever atom you choose, as far as it's in the same plane, the distance they're traveling is exactly the same. So this is the famous Bragg condition. And now but drawing this plane a lot of time is quite tiring. So what we want to do is to simplify with what we have drawn. And now if you draw a figure like this, you have a sphere where the radius is inverse of the wavelength. And we had defined the reciprocal vector where the length is 1 over d, and which is perpendicular to the plane of the crystallographic lattice. So then actually when the sphere touches this point, if you look at the geometry, it is exactly the same as this condition, because this distance, for example, this is 1 over d multiplied by sine theta. Okay, so and this length is twice it. So this condition of drawing this triangle is exactly the same as this Bragg condition that you can just change this order. So then what if you have a sample, you draw where is your reciprocal space, reciprocal vector in space, and you imagine a sphere shining from the that goes where the origin is on the X-ray beam, and the sphere touches the sample, and you imagine when where the sphere is touching this reciprocal vector. So this is an example we had. So these are done at KTH. So they had a series of nanopapers made of hollow cellulose. So they prepare hollow cellulose and disperse in water and make nanopapers, or some with enzymatic pretreatment, or with tempo cellulose. When you look with a beam perpendicular to the film, they look relatively isotropic. They have intensity about the same in all directions, which is expected. But if you look at the intensity of the two rings, for example, two rings here, hollow cellulose and enzymatic, it's classical for cellulose 1. It has similar intensity ratio between the two rings. But if you look at tempo cellulose, the intensity ratio is different. Now, when you look at the X-ray, the side view with the X-ray parallel to the film, this is what you obtain. So you have some oriented pattern, which is typical for those two, which is typical intensity ratio. Again, if you look at in this direction, you have the intensity, by the way, which looks very much like what you saw for the primary cell wall in the grazing incident. It's almost exactly the same intensity distribution. In the case of tempo, you will see some difference, and especially you see this relatively sharp arc here, which you don't see in other pattern. So I was quite excited by this pattern and try to explain that it's so interesting, and actually it's quite difficult to let people understand that it's interesting. Also, there's something. So what I'm trying to explain is what happens when you have planar orientation, which is the following. So you consider, so this is, I'm considering now a lot of points around the fiber axis. So if you have a cellulose fiber that is rotating around the chain axis, the diffraction of, for example, take 200, something that is perpendicular to the fiber axis, is along the circle. Okay, if you have a random orientation and you have a lot of this in one direction, you will have this kind of situation. Now, you rotate it on another axis. You have the fiber which is lying on a plane, and this fiber itself is orienting random along the axis. And then you have this kind of situation where you had a point, and then you rotate this point on the axis perpendicular to this. And this is a typical case of planar orientation, where in most of the case where you have cellulose fibers, you have the fiber chain axis, which is random oriented in the plane, but this is restrained in the plane. So then what happens is that you have high concentration of this point on the pole. And actually, if you have perfect orientation in the plane, then this intensity is diverging to infinity. So then you can simulate this once you have distribution of you assume a distribution how much the chain is oriented out of the plane, you can simulate what would be the orientation of other axis, other diffraction, like so here it is a case of uniaxial orientation where you see the distribution of the chain axis in one direction, and then you have the equatorial refraction like 200, 90 degree to the orientation. This is the typical case for polar cellulose and enzymatic pattern, where you see this strong diffraction in the 90 degree. But then you can also have a case where, like in the case of Wallonia cell wall, where you have one crystallographic axis which is preferentially oriented with respect to the fin plane, and then you here it's much less visible than the case, because as you see, the intensity becomes much smaller due to the fact that it is diluted by the fact that it is rotating randomly around this axis. So visually, you see that this is very faint, and you see it much better on the 2D images. If you look, what I did is to look at the orientation, the intensity distribution around this circle, so that I will capture this 1,1,0 arc. But actually, when you reduce into the intensity profile like this green one, you see a small faint peak, but it's much less, and this is the intensity is much smaller because of the fact that you are diluting by this circular distribution around an axis, where you have, when you have the diffraction which is oriented perfectly in this direction, you don't have this dilution, and the intensity will be much stronger. And sometimes, so in the same series, we did also diffractometry, because what I saw here in this pattern is that you see a strong intensity here, which I suspected that it comes from the continuous X-ray of because we don't have monochromator in our X-ray system. And if you do go to the powder diffractometer, they are all usually equipped with a monochromator. So we can get rid of the effect. And furthermore, we emphasize something which is perpendicular to the film. If you have a film, actually what reflection mode diffractometer is doing is only tracing the intensity along the vertical line of this one. So you are accentuating all the aspect which is in this direction. And because what we are looking at is this peak in the direction perpendicular to the film. This is in this case, it is well adapted. And now we see a very strong peak in this small angle, which probably comes from the layered structure of the microfiber itself. So this is the distance between the microfiber in the layers. So this is the drawing of what we deduce from the series of measurement. Very simple measurement, because it's just 6 exposure, each of them maybe 30 minutes in this small apparatus. And what we can deduce is, so if you have a crystallographic plane colored with different color as shown here. In the case of hollow cellulose and enzymatic cellulose, the chain axis is random, and also the surface exposing on the film is roughly random. It's all of this. And it is also the case of primary cell wall, because the X-ray pattern we see is the same. In the case of tempo, for some reason, this plane is over representing in the system. So you have the chain axis is random in the plane, but you have this 1 bar 0 plane, which is predominantly parallel to the surface of the plane. So, which is similar to this situation where you have this peak. By the way, you see that because I knew this picture, when I saw this picture, I saw it's quite similar to the Valonia, and you see that you have stronger at 45 degrees. You see that 200 is stronger than the other two scattering. And you see a weak 110, which is exactly the same as what you see here. So this was for series 1, but also you have similar orientation, uniplane orientation in cellophane, for example, regenerated cellulose in general. Again, this is very old paper in 1940, and if you're interested in texture of any sort, I strongly recommend to read this, because it is explaining a lot of things on regenerated cellulose and texture inside. And here, this example is just cellophane. If you shine X-ray parallel to the film, you get this kind of pattern. So this spacing is the distance between. So you have a flat pyranose ring, and in the case of cellulose 2, they stack first with hydrophobic plane, which is distanced at about 4 angstrom, and then you have this the distance, which is the distance between the stack of hydrophobic sheets, which is now parallel to the film. So this is directly related to the behavior of cellophane, because cellophane swells a lot in water, but only in the thickness direction. So that's exactly because the water will penetrate only in the direction where you have hydroxyl groups, which is perpendicular to the film. And this photograph, just simple photograph, illustrates directly this. So this peak, which is at smaller distance from the center, means that you have larger spacing. Okay, and this is the spacing between hydrophobically stacked antiparallel cellulose sheet. And water can more or less penetrate inside, because if you have wet sample before completely drying, you see this peak is at lower angle, and when you dry, it will appear stronger and at higher angle. And here, if you look with the beam perpendicular to the film, now you don't see this peak at all, because you don't have any of the crystallites in the condition where you're filling the Bragg condition. He can also rotate the film during the exposure, and then he gets this kind of pattern, which is the same as just putting rayon fibers. Because well, it's a question of the size of the beam and the size of the fibers. If you have much smaller beam than the fibers, then you would see this in the skin. But if you just do the X-ray experiment in a lab, you don't see this internal structure of the fiber, and then it is averaging out over the whole fiber. So I will just, this is another very interesting experiment, again, very simple, a few exposure to see in which direction chitin is spin in a tube worm. It is done by Yu Ogawa that you might know about 10 years ago, and it is very little cited, but it's very interesting. So tube worm is you find in deep sea seabed, a kind of worm that has a chitin shell that has this form. You have this kind of trumpet shape, and this is the bottom, and this is the top of and the worm is living inside this tube. And so we look at the structure by shining the specimen from different direction. And from the top, okay, so in the cross-sectional view, we obtain this. And this directly tells us how already that how it has very particular orientation, and it tells us the direction of the reducing end in the structure itself, because it is asymmetric. If it was just one plane with respect to the membrane and having up and down microfibers, then you should have a pattern that is symmetrical along the vertical axis. Now you have a pattern that is completely asymmetric around the vertical axis, which means that all microfibers are pointing in one direction. And when I say one direction, it means that the reducing end is always, in this case, from top to bottom of the tube. And now we cut it from, if we shine X-ray from this direction, we have an Ewald sphere which looks like this red. If we shine X-ray from this direction, you have Ewald sphere like this. So please remember that here you have strong, here you have strong pattern, okay? And now you see the pattern when we have the X-ray parallel to the membrane, and this is perpendicular to the membrane. And then you see that on the right-hand side we have strong reflection, and here on the left-hand side we have the strong reflection, because we are just cutting here. So we see that we cut this part where you have a lot of this reciprocal vector pointing. We are cutting here, and we see this strong reflection. So if you imagine the system with a reciprocal vector concentration in reciprocal space, the interpretation of this kind of pattern becomes very straightforward. And you also see that if with the X-ray beam parallel to the membrane, in both cases, the z-axis is roughly vertical. The z-axis is roughly vertical, but you have more of the misalignment if you look at with the X-ray perpendicular to the film. So you have different aspects of the orientation. But main point here was that we can directly deduce the chain polarity just from a few X-ray diffraction. And then, so those, up to now, those are all done in a lab X-ray. You don't need really sophisticated X-ray source. You just expose for 30 minutes, 1 hour, and you get very nice pattern like this. So here, nowadays, you have a lot of availability of synchrotron. You have Formax in Lund in Sweden. So then you want to have something that justifies the use of high brilliant X-ray. And this was one example like this. Yeah, the way we analyze it at the end is always the same. But here what we did is to observe what happens during the drying of nanopapers. So you have, in this case, so Leng One at that time in KTH, he mixed clay and marmorinite, in this case marmorinite and cellulose nanofibers, made filtered, and then you have a wet cake. And we just put this wet cake under the beam and just scan over time what happens to the sample during the drying. So we have two samples, one which is the cake parallel to the beam, and another one perpendicular to the beam. We just put it on the sample holder and we scan over and over several hours. So here we see we can see several things. So here in this case, we see with a beam perpendicular to the film, we capture isotropic scattering of water always, and which decreases with time. So we can just follow from the intensity of water scattering, you can more or less deduce the water content with respect to the cellulose scattering. And here also measured in parallel by just weighing in another system that gives roughly the same evaporation behavior. So you know at each instant how much water you have in the system. With the X-ray beam perpendicular parallel to the mat, you will get this kind of oriented pattern. So you can retrieve the peak position, how it is aligned, the intensity, and so on. And very often people tend to do integration because there are softwares to do just azimuthal integration of intensity. But then you will lose information because CNF diffraction and marmorinite, they don't have the same azimuthal intensity distribution. So if you just integrate along the whole azimuthal axis, you will lose this information. So what we did is to try to fit all the data points directly into the dimension with a function to deduce different parameters. So yeah, I forgot one slide, but anyway, so we can retrieve a lot of information that's, if you look at directly from the 2D image and try to figure out what kind of parameter put in it, we get more information than if you average out the intensity along this azimuthal angle. So in conclusion, I that X-ray scattering, even the simplest setup, is a very powerful tool to analyze textured system. And it sometimes it's very complex with many potential parameters, and it's very important to directly appreciate the scattering pattern before doing any numerical analysis. There are a lot of so people tend to jump into the analysis of integration and peak fitting or some scattering intensity fitting, but just the direct two-dimension image is telling a lot of history of the sample. So I hope I've convinced you on this. And thank you very much for your attention.
Thank you a lot. There was a lot of information there. If you look around, is it one conclusion that you can see very often that people go directly to integration and don't care so much about the 2D images? Is that high risk then?
Yes, because you can, well, you have a lot of the information in 2D is very rich depending on how it is done, and you reduce it into one-dimensional problem. So yeah, for example, if you just average this one, you will completely lose the texture, and you don't understand how it is oriented and why the intensity is different. If you look at different direction, look at the 2D pattern, you don't even need to calculate. The image is directly telling you.
Yes, absolutely. Normally we open up for questions, Josephine. Per, do you see any questions in the chat? No? If you have any questions.
Yes, we have also raising her hand.
Yeah, what's that? I allow you to talk, so you can say it. I hope.
Our newest fiber PhD
Hello.
students.
Yes, hello. Can you hear me?
Yes.
Absolutely.
Okay, thank you so much for this nice talk. I read this paper on the like different types of cellulose and how they, what's it called, like uniplanar stuff with great interest, but it's very difficult to wrap your head around, I think. And you mentioned now that when you look in the perpendicular direction, you have like with the tempo cellulose, you have a different ratio between the 200 and this like 110 1 bar 10 peaks. Like there's a different ratio there. But why is that? Because I find that very difficult to understand. Is there different amounts of those planes present in the?
Yes, so yes, because you see, when you do X-ray shining perpendicular to the film, what you're doing is actually cutting this plane like this. So you're looking at the intensity here in this direction. So actually you see this one very strong, okay, because your Ewald sphere is cutting here. You are completely neglecting those.
So you do see some kind of, like you see a cut, like the orientation impacts also this top
Yes,
view
so
data.
imagine in 3D the distribution of the reciprocal points, and your Ewald sphere is cutting one, well, yeah, the sphere. So when in this case you have very little reciprocal points, but the sum of them you see is dominated by this one, 110, and which appears strong in this figure. And you see much less the 200 because you see the edge of this 200 distribution, but only the edge. So at the end, they look very similar in intensity. Here you are cutting here, and because you don't see preferential orientation, they are all weak. And it's weak because it's thin, but also it's because it's rare. And you see the 004, which appears actually strong in this pattern.
Okay, so it's like I was imagining, I think that the top view data wouldn't be influenced by the texture because the texture exists in the side view, but actually the side view
Everything
texture.
matters. Yes, yes.
Yeah.
You just perceive it as an isotropic scattering pattern, but because you're cutting with a symmetry, with something that is centered on the symmetry axis.
Okay.
It happened to look symmetric.
Yes.
But just because you're cutting with a specific axis.
Okay. Thank you. That helps me a lot.
Thank you, Åsa.
Yeah, that was my question. Thanks.
So Åsa defended her thesis last week.
Yes.
So,
On Thursday.
yeah. Do we have more questions here? No? Okay. Per, do you have any?
So yeah, first I was coming to or felt that your presentation was leaning towards we don't really need synchrotrons and all those things, but in the end you finally came there. And
I mean,
when you have the best possible beams and everything, and you showed the slide where you could estimate the moisture content, or is that a relative moisture content or,
mean, you
or would you
can-
dare?
As far as you scale everything in absolute, you can, if you have the measurement of the transmission X-ray, then the intensity you directly measure is proportional to the quantity you have. So you can have the mass of water in the beam directly.
Yeah. So my question is basically, do you dare to say that you don't have any water left, or is it more on a relative scale that you have a little or much water?
When it's dry. I mean,
can you
okay.
can you use X-ray to say that it's
No, because
absolute?
what you see, okay, so when you have bulk water, you have the typical scattering pattern of bulk water. If you don't have bulk water, we actually X-ray does not distinguish any chemistry. We are looking at only the electron density fluctuation. So if you don't have this typical structure, then you don't see it. But so this is when you have, so it is only valid if you are drying from very wet cake to roughly dry condition that you lose bulk water. But I'm not saying that the end 5% we can detect by scattering, because then the structure would be different, of course.
Okay, that was a pity.
Yeah.
We have a hand raised by Lars Vågberg.
Mm?
So Lars, you're allowed to talk, Lars.
So, can you hear me now?
Yes.
Yes, thank you, Yoshi. This is not my field of expertise, but I've been on the same token as Per was mentioning. The last couple of weeks, I've been trying to understand how water is associated with cellulose, and then you always run into this existence of the hydrophobic planes that you mentioned for the regenerated cellulose specifically. I ran into a paper where they had calculated the surface energy for different orientations of the cellulose, and I was astonished by the high surface energy they had calculated. Do you know of any? Have you done that kind of calculations yourself, or do you know of any reliable estimates? Because I'm a bit allergic to hydrophobic, hydrophilic. I would like to know the grayscales. How high is the surface energy in different planes? Do you know of any calculations on that?
I don't know what kind of, because you mean surface energy in terms of if you split them in vacuum and how much energy you need to split the surface. That's what you call it.
What I mean is the concept of surface energy. I mean,
well,
if you,
I think
you
usually the concept of surface energy is you split the two, expose the two surface in vacuum and see how much energy you have to input to split the two to put it in the very long distance. Right?
Yeah, but the surface energy is the work needed to split the surface, but it's also a measure of how the surface is interacting with other materials. So if you have the surface energy, you can predict the work of adhesion between the surface and water, for instance. But if you say hydrophobic, it's basically 0 or 1. So I'm missing the grayscales.
Well, okay, when we say hydrophobic, it's just because you have only CH exposed to the surface, CHO, the easy oxygen, which is different from having OH on the surface, right? So,
but it's a rather useless. It's a rather useless statement if you cannot quantify it. I'm not blaming you, no. I'm just curious. I would like to know.
So,
how hydrophobic is hydrophobic? Is it is a contact angle 125 degrees or 28 like Teflon, or is it 115 degrees, or what is it?
okay, so I think the concept here, it's not that to expose it, but in general, when you make cellophane, you dissolve in a kind of aqueous system and regenerate in aqueous system. So the structure, it is the first molecular association is due to the hydrophobic interaction, and it is never meant to be exposed to other stuff during the regeneration. And it's just the growth direction of the structure in this regeneration process is what along the direction of what we call hydrophobic interaction. And so as you see in the case of cellophane, it's really strong because, and it's always what we see in regenerated cellulose because it is really predominantly growing in that direction.
No, I have identified this as an area that needs more investigations and quantification because it's an interesting concept. But today it's more, we are using it more to say on or off. But I understand fully what you're saying with cellophane, of course. That's no problem with that.
Yeah, and of course, people try to, like Yamane, Professor Yamane, you know, that he tried to expose the other surface to turn the molecule 190 around the axis to have the other surface exposed, and then you would expect to have hydrophobic material. But that has never been achieved. So.
No, but I mean, this can be a subtle balance. It's hydrophobic is usually when the contact angle is larger than 90 degrees. But if you have a contact angle that is 10 degrees and now one that is 65 degrees, you have a big difference also.
Yes, the point is that people never achieved the orientation of having the other molecular surface exposed on the surface. So you cannot measure because you don't have the material.
No, but people can do everything with molecular dynamics modeling today,
course,
so they should be able to do that. Yes.
Experimentally, it is not achieved.
No, that I can buy. But I was more. You've been looking into this. I was more. If you had some reliable references, I would have really enjoyed them.
No, but my preference is to have some experimental parallel. So it would be nice to have a way to achieve this.
Yes.
So far, nobody.
No, for instance, we have another thing that would be interesting and links to what Per was saying also is that if you're heating cellulose for 130 degrees for a while, you will change the contact angle from around 40 to 90.
Yes.
And nobody knows why that is. That's So I mean, but we can continue this discussion later. But I just wanted to raise it because I jumped. You were online and I had the opportunity to ask.
Okay, but yeah.
Yeah, that is a very interesting question. But I see that we have time. We are running out of time. So I think it is should we end now? We are 4 minutes late. I suppose it is.
There's one more hand, so the choice is it a quick question or should we?
Yeah, who is it?
Yeah.
Please go ahead.
Hello, Yoshi. This is Alireza from Mid-Sweden University. I have two questions about based on your experience on scattering and the diffraction on this, I would say, different semi-crystalline polysaccharides. And the first question is how big can be the crystallite size of this semi-crystalline polysaccharides based on your experience? And the second question is about what is your thought about the influence of the defects, and do we have any dislocations, I would say, that is common in the polycrystalline material in the semi-crystalline polysaccharides?
So it's a very vast question because in the case of, are you talking about when you crystallize your polysaccharide or in nature in general? Because in the case,
yeah, please.
Yes, in the case of native cellulose, it is more or less dictated by how it is synthesized in the first place. So because the crystallization and the synthesis is almost simultaneous. Once you have a polymer, it's very difficult, and if you have a dissolved state, it's very difficult to align them and crystallize, because you have a frozen state. You cannot extend the whole molecule, and if you want to make a crystal, you have to extend it. So that's not happening with cellulose, and it is more controlled by the instance it is polymerized. And then if you want to recrystallize, it's much better to work with oligomers, and then you can grow larger crystals. And in principle, if you have monodisperse oligomer, you can grow into large crystals, but this is standard crystallization problem.
Mm-hmm. And do you think that can it be identify any defects or the dislocations that is common in the polycrystalline material by the diffraction technique for this?
Okay.
Yes.
Yes, we sometimes, in a very special case, like if I don't know if you know paramylon, which is a beta 1,3 glucan. In this case, we see some streaks, probably due to the dislocation of certain plane, like in what you have in an inorganic system. But to see this kind of things, you need a fairly organized system. So it's rather rare, because most of the polysaccharide system, it's very little crystalline. But in some case, yes.
So it's possible to have it in the semi-crystalline material.
Yes, if you look at some exotic sample.
Yeah, and the last question, what is your thought about the widely utilized Scherer equation for the crystalline size estimation of the cellulosic material, for instance? Is it correct?
It's mathematically correct. It depends on how many digits you want to, because roughly 5 nanometer, roughly 10 nanometer, that's fine.
Yeah.
You don't deviate too much from other measurements.
Yeah.
But if you want to say 2.56, that's when the problem of where do you define the end of the crystal. So mathematically it's fine, but it does not have that much meaning, right?
Yeah, that's true. Thank you so much for the inspiring talk.
Yeah, I fully agree. Thank you a lot. But I think we should end now, and I hope that we can see you in person soon and continue the discussions. Thank you, Yoshi.
Thank you.
Thank you.
Thank you.
By the way, can I ask you, so the 130-degree stuff, when you re-soak in water, then it becomes back, the contact angle comes back after some time?
I guess that's Lars has that probably more on top of his head, but as I understand it, very, very slowly.
I haven't done the measurements, but it's a good question. But then you need to moisten the samples. I mean, I think has a lot to do with removal of water and what is changing the structure then. But I don't think it's a surface structure problem. But
yeah, but it's just you looking at the instantaneous contact angle, or it is just staying over a long time?
No, this is after I think it's after 10 seconds, but they are rather stable at that time. So we waited for them to reach a stable level. It's not the initial one. It's not the initial one. No.
Okay.
Will you come to Stockholm, Yoshi, to the BVS conference?
Not this one, but I'm coming for EPNO and stop by Stockholm.
Okay. So in Sundsvall, yeah. Okay. Nice.
But dwelling on that topic, if one heat under X-ray radiation, would a restructuring of the polymer itself show up?
Yes, in a sense, yes, I think so. But yes, of course. But you might not want to do that at synchrotron because you will hurt the sample, or you have to change the place where you take the scattering.
So then basically there are experiments to discuss
Yeah.
at some future occasion.
Yes.
That sounds very good. I need to leave now, everyone, but I look forward to meet you next time.
Thank you.
Yeah.
Okay. Bye-bye.
Have a good day, everyone.
Bye.
Bye.
Recording stopped.